other, we're still going to pass the mic around to make sure that it's an inclusive meeting. For those of you that are joining virtually, we are tracking your questions. Please join, write your comments, questions. We'd love to welcome them. And so this is a good practice, the session, breakout, making sure that you're in the right breakout session. This is the breakout session for good practice in building organ-level atlases that are going to be moderated by Kerstin and myself. So I'll let Kerstin introduce herself.
My name is Kerstin Meyer. I'm the Working Science Institute in Cambridge in the UK and part of Sarah Teichmann's group. And we've mostly been me in particular, we've mostly been contributing to the lung data sets. And of course, you've all heard quite a lot about that from Fabian earlier, from Malta earlier. And of course, some of you might have seen in the program that Martijn Naveen was supposed to be here today, who's done a fantastic job in coordinating the lung cell atlas. Unfortunately, he has COVID today, so I'm standing in for him. But I think I will, on various occasions, reach out to the people from the Lung Cell Atlas community, because I think that's one of the examples where we're actually already quite advanced in terms of the atlas building, but hopefully we'll get a good discussion with all the other organ atlases as well.
Chloe Villani. I had to also step in for someone else who couldn't make it this morning, so here I am again. Although I was supposed to moderate that session. This was my one responsibility for today. I'm one of the coordinator of the Immune Bio Network. And so the scope of this session here is to try to see how we can help each other across bio networks. Some of us have found solutions. Some of us are a bit more ahead in our roadmaps than others. And so there's a lot of things that are probably generalizable, or we may realize that we're all encountering the same challenges and we're not realizing it. And so before we, Kerstin and I tried to organize a PowerPoint around some of the key themes on the topic of building our roadmaps. A lot of us had to reflect on this over the course of the last 2 months as a community. And so we want to hear from you, what are the challenges and opportunities. And so we're going to try to use this as a whiteboard and type the notes and your feedback as we go so that also folks online can follow. Just so we get a sense of who's here, I love to kind of hear who's here from which bio network. So I have the list here on my phone. So who's from the, going in order, is there anyone from the adipose bio network? Yay, represent. And then breast, do you have breast? It's okay, we'll cover breast. Development?
I'm sort of.
Sort of, there you go. Eye? Okay, genetic diversity? Okay, thank you. Gut? Yay, heart? Immune? Oh, immune. To be fair, it's a question of proportion and distribution. The bio network is quite large for immune. Kidney. Oh, there's a lot of kidney. Liver. Thank you. Lung. Yeah. Oh, there's good lung enrichment on this side. Musculoskeletal system. Nervous system. Oral and craniofacial. Thank you. Organoids, pancreas. Oh, reproduction. That's good. And skin. Great. Did I miss anyone? Any bio network emerging? Great. So we actually have a fairly good representation. Thank you all for joining. And so so we have technology groups, yes, perfect. And I guess we have representative of the computational working group. I'm looking at you. Yeah, well, can own it, Oh yeah, perfect. And so this is tiny. Is there a way that I can make this bigger? No? Well, I mean, not on the computer. Is there a way, sir, that we can make this bigger? I'm not that, okay. So I think I introduced this already. We're trying to kind of get a sense of where things are for everyone in their bio network. So we have an hour and a half to cover different questions. And would love to hear thoughts. Yeah, I kind of skipped the summary here because I think we already kind of mentioned this, right? we're all drafting our roadmaps. We all are facing different challenges and sharing some opportunities. So the first sets of questions are related to on building the first version of our atlas, which, in principle, I was speaking with some of you yesterday night. In principle, sounds pretty easy in concept. In reality, I can speak for myself, for the Immune Bionetwork, it's not that trivial to get our data sets and figuring them out. And so love to hear your thoughts, and we're going to pass the mic around, and we're going to try to type notes at the same time. We have 2 dedicated scribes as well, so gathering everyone's thoughts. So love to hear about how different groups are currently identifying and prioritizing which data sets are going to go into version 1.0. Do you have any recommendations in terms of how to coordinate, how to prioritize, what modality to prioritize, knowing that we are supposed to be releasing a draft atlas of our respective bionetwork by October 2023. It sounds far away, but it's not, because it means you kind of need your data set now to be able to QC and start, working as a team. So, any thoughts? Yes. Wait, Mike. And if you don't mind mentioning the bionetwork you're representing.
Lab at the Sanger Institute. Actually, I'm across a number of bionetworks, lung, immune, and gut. So the point that I was going to raise is before considering any kind of data types, modalities, etc., it's really a challenge to get, because to integrate large, multiple data sets, you need the raw data. And so I think that's a real challenge for access-controlled data, which, of course, there's reasons for that, but it's a huge administrative challenge to get raw data in some cases.
Yeah, no, I think we all recognize that, and that is a problem for all the data sets. Luckily, for where we have organ donor data, we have, of course, got open access, which makes it much, much easier to share. But I think, Amanda, what you're bringing up there is actually a really fundamental question about the community, actually, that before you can start to build an atlas, you really, really need to have a community of people who all come together and who are all willing to put in time and put in effort. And again, I want to give a big shout out here to Martijn Naveen and Sasha Misharan, who've done this coordination for the lung cell atlas. And I think it's really been through their interactions with the community and the fact that they've, reached out to people, the fact that the community trusted them with their data, that we've actually been able to make a lot of progress for the lung cell atlas. And I think a part of that community building is also having funding, having money to bring people together so that they really get to know each other. And of course, for lung, we had the Discover in Europe, and then also the CZI Lung Seed Network, which was international but more US-based. So, that community building, I think, is really important, because also the community needs to then put in time and effort when they're requested to do that. And I'm sure this is a theme that'll come up again and again. So I'll stop there for the moment.
Well, don't we have 2 mics? There we go.
Matthias Gratzler, University of Michigan, Standard Working Group and Kidney. Yes, I think this is really important. And please don't reinvent the wheel. There are, with DUAs, MTAs, and yes, those ugly words have to be said, because without them, no tissue or data will travel between institutions and continents. And several of the working groups have established frameworks which at least minimize the administrative burden and delay to share tissues. And what the Kidney Network found very helpful, that we have one reference tissue source who is shared between the HCA Seed Network, HubMap, and KPMP. So there is one benchmarking tissue source, which hopefully, when we are talking later on the version 2 and the integration of multimodal data sets, could become quite helpful. But that requires that, yes, there is to one entity, and in my group I have a full-time FTE who does nothing else than pushing these paperworks around, because usually your institutions are only so responsive if they don't get a reminder and some pre-populated forms. So there are formal frameworks available to facilitate that if you're starting your work anew. And please build in time for that, because this will require several months usually to get these systems in place before you can share tissue and data widely.
May I ask how you ended up funding and supporting that full-time FTE?
So this is coming from, to some extent from KPMP and to some extent from discretionary funding, because this clearly was something where we all felt it's absolutely instrumental to moving this project forward.
I think a lot of us are experiencing this, and it's challenging to obviously by version 1.0 we kind of need to make it happen even if you don't have support right now, but I think that's something that's coming up across bionetworks. We're realizing that a lot of us are not funded to try to gather these data.
Absolutely.
And so it's all after hour.
Absolutely. And this is and the final comment in that area. At the same time, particularly if you are harvesting tissue from study participants, obviously talk with the ethics standards working group so that you get adequate consents, which allows you to share the samples broadly and effectively, because nothing more aggravating than if you have nice data and the data cannot be traveling where it should be.
There's two and then...
Hi, I'm Zorina Gallus. I'm one of the coordinators, the NIH Hub Map, so I'm not part of the SEED network, but I think we face similar challenges, and a lot of the investigators are shared across various funding opportunities. So my question is more fundamental. How do you sample to actually say that you have an atlas of an organ? I assume that every organ has its peculiarity. So is there a strategy? I would love to know if there is a strategy on sampling various organs to say that you actually have an organ map.
If you don't mind holding your question, we have a whole slide about this, because I think one of the thing to consider at this point in time is that for version 1.0 we have to go with what we have. Yeah. Right? Whatever we have now is what's going to be in version 1.0, and so we need to. Yeah, the tissue per organ system, which is going to be likely only one or two modalities, likely very sparse, and we need to figure out the data gaps. So we have a few slides coming up about the data gap, tissue sampling, and if about 40 minutes we haven't answered your questions, please raise your hand again, okay? Christoph.
Speaking of MTAs and data release and all of these things, it seems that at least a quasi-consensus in Europe is that the raw data, the sequence data, are identifying and under full control and with very strong restrictions, but the count matrices are not. The count matrices are considered non-identifiable information, meaning they can be passed around entirely freely. Do we think we can technically, without compromising too much of quality, build integrative atlases solely on count matrices?
That's a good question. We have someone that may want to offer thoughts here.
Malte Luken from Helmholtz Munich. I think this is a really good point. What we did for the lung actually is that we distributed a raw data processing pipeline, so we have some consensus there, and all the individual kind of data contributors committed under the supervision of Martijn and Sasha to run these pipelines. So we had some kind of consistency there, and then only worked with the count data moving forward. Of course, what's crucial here is how much metadata can you share, right? On the one hand, we can share the count data really well, but metadata resolution in some countries is differently identifiable. And I think that's an additional question that is a little bit more difficult to answer and sometimes leads to limitations. Sorry?
Where is metadata, sample-assisted metadata actually?
So actually, Kerstin was a great example where I think age ranges could only be shared.
So, for example, in Cambridge, we have organ donors, and there aren't a million of them, right? It's a relatively small number. So if we give the exact age, then our ethics committee felt that they were almost identifiable. So in the end, we had to give age ranges of bins of 5 years. Again, from a scientific point of view, of course, you'd like the exact age, be better, but I think that's a good compromise.
Just to follow up on what you said, it sounds then, would you think the consensus is that we really have to treat them all the same and run them through the same pipeline, or it would be actually all right, since we have so many other confounding variables like, from age to different body sites and so on, so that those are actually much worse, so to say, than the count matrix, how it was pre-processed?
Oh yeah, I'm coming back.
Yeah, sorry, just to address that quickly. I think it's actually crucial to have batch variation, so have batch heterogeneity, so otherwise the models that you train, as I mentioned earlier as well, are not necessarily fit to map new data on top of, unless you have some biodiversity there. So I'm not too worried as long as, like, I can't talk for the data generators who have each their own optimized protocols for their particular questions, right? So obviously these protocols should not give you terrible data, but I'm assuming that is optimized. I cannot control that. So actually there, like, whatever decisions were made by the community to generate optimal data is probably also the decisions that will be made for new data be mapped on top. So if that diversity is captured, I think that's suitable. I hope that answered your question. I'm not sure if
And I mean, actually, in terms of building atlases, it's really encouraging to hear that having lots of diversity improves the atlas quality rather than detracts from it, because of course what we're really after at the end of the day is the biological variation, which comes out better if you have different profiling modalities, let's say 3', 5', what have you. Yeah.
Hi, Alex Bernier here. I'm based at the Center of Genomics and Policy, and we actually coordinate a lot of the ethics working group's work in essentially establishing, common standards for biomedical research ethics, norm compliance, and some of the data governance. One thing I wanted to chime in with is that we have a set of tools that sort of set out things like standards for informed consent materials and for assessing in collaboration with your research ethics committee, your institution, how data can be shared, both in terms of how openly and then also what kind of practices, procedures to put into place. The other thing I'd like to add is we have, and it's available on the website, there's a sort of an email helpline. So for kind of ad hoc help with kind of navigating some of those data sharing questions, of course we don't dive into any of the legal parts, so the GDPR, things like that, but really more from a research ethics standpoint, we're happy to kind of field case-specific questions if anyone needs support on those efforts. One thing I will say with regard to the general discussion that's going on about, what can and can't be shared both openly and then in a more controlled manner and then at all. So the HCA, of course, is in the process of sort of finalizing its own both managed access and open access data standards, and those are being made publicly available. But from a more general standpoint, we talk about things like identifiability and what can be shared, what can't be shared in absolutes. In reality, there's two big things that make it very contextual to determine. The first one is, of course, that identifiability regulators, they tend not to look at it just looking at the data. They tend to consider also the data sharing practices intended, how many people are going to access the data, whether you have things like auditing practices and contracts in place in determining how data can be used. So that means that something you might think of as very controlled, very identifiable in a particular setting might actually be more free to use if you make the case that, hey, because of the measures we have in place, it's less high risk. And the other thing is that different jurisdictions, which is, I guess, a blessing and a curse, are coming to really different standards and approaches. So sometimes, even if you look in the EU, some member states, the regulators are very stern on the movement of things like genetic data, but other member states, there's a little bit more flexibility. And I think it's something that as a scientific community, we kind of have an obligation to sort of, set standards internally and say, look, these practices are really important to us, and so regulators need to align to those. I think it's really great that we're having the discussion. Thanks.
This is Bruce Arno, Cincinnati Children's, and I'm in a couple of different networks. Just with regards to the matrix sharing, the problem with that is it's really inner join, outer join, and what was the workflow that generated the count matrix, and how much we don't have good metadata that goes with our count matrix distributions about what was the provenance of the workflow that led to that these counts summarized. And when you're at the fringe of, and you know, to help discovery efforts and, you know, people who are, you know, kind of leading different charges into you know, fine details, like, you know, let's say link RNA function or other non-coding RNA function. These are very dependent on the version of the genome that you're working in, the transcriptome library that you're working on. And some of those can even be, you know, very sensitive to what the cutoffs were on the aligners and that sort of thing. So that's the stuff that we, you know, you want to be able to control, and you want to be able, and then you want to be able to have a new, clean take over, you know, a new reference atlas. And as we get new, better human genome annotation, and then see ethnic diversity, and then, you know, new genes that are forming or duplicated or pseudogenized in different populations, that's all important to then realign the transcriptome space over that. And so that's why, you know, we need to do this in, you know, Providence.
But I think, you know, Malta's point there, that if there is a community that, and a computational lab that leads the integration, that, you know, has expertise in really putting together a really efficient workflow, and that then gets redistributed back out to the community. And that's really where we do need that community to then actually run the algorithm and put in the time and feed the data back. But I think the lung cell atlas has shown that can work. But, you know, it might also be good. So we have had a few examples of the human lung cell atlas where people have already come together and donated, not donated, but given data into the system. Maybe we can hear from one or two people who are still really trying to pull things together where they're finding it's not really working, and just hearing what the main challenges are.
Yes.
I just wanted to add something on. So Maria Kasper from the Skin Network. Have any one of you already used like data anonymization, such as Bamboozle? So there are now programs out, for example, from the Sandberg lab, where they actually have algorithms where one anonymizes data, like RNA sequencing data. They take away all the person-related sensitive information, such as SNPs or indels and so on. And you basically would be able that way to share, and you get the raw data. You just take out the SNPs, and you still could align it to your favorite. And it's something like for maybe the community to check, or even from the law side, whether that is something we really all can use, and then openly share the data, like raw data that's anonymized.
Maria, let me do that. I completely agree with you. And in fact, I published a data set about 5 years ago, a single cell data set with Fluidigm, that where we wanted to share the data, and we just reduced everything to the reference reads and then put out the FASTQs in reference space, like 3 years before Bamboozle. but perfect. Yes. So this is a really cool point. And so can we commit to putting out FASTQs, a transformed, de-identified FASTQ that's, you know, that really is adequate for reanalysis against a new reference genome? I think it's a really strong thing. Let's do it.
I think that's a good point, although I will say that the genetic diversity working group may find this a little challenging since one of their primary goal is to look at this, but I realize that a lot of the donors didn't consent for this, so perhaps it can be like a workflow depending on what has been consented.
The IRB should be able to say that
yeah.
this is a method.
Yeah, so it can make me like very clear. Yeah. To build on Kerstin's point, we were hoping to hear, and I can step in as the Immune Network coordinators. We wanted to hear from those of you that are trying to gather data sets. All of you already gathered your data sets for version 1.0, and if you haven't, want to hear from you and see what are the challenges you're facing, and perhaps others can give some feedback.
Hi, so I'm Margot from the Adipose Network. As maybe you might have noticed yesterday, we're still trying to get people to contribute to the Adipose Network. Part of the problem is, like I was saying, in order to get all of the cells, including mature adipocytes, you need to do single nuclear, which is a pain. It was a really pain for us to optimize and get good data from that. And I believe it's a pain for anybody else who's working on adipose. So right now, there are very few adipose data sets, and even fewer if you're counting single nuclear data sets that have the mature adipocytes. So one of our problem is just that not very many data sets exist. And then our other problem is that of the ones that do exist, many of them are different adipose depots because different labs are interested in different adipose depots, which have different metabolic functions, basically. And so, we've been looking at white fat, but there are other groups that are looking at thermogenic fat. And so do we combine those into one integrated adipose data set? One thing I don't know if we're going to talk about later is how do we integrate that when two labs are doing such different experiments that you want to you want to reduce the batch effect between labs, but you don't want to integrate over the actual functional differences between the different depots that we're looking at. So that's something that we're.
Bruce.
So is the fundamental problem that you're talking about differential capture efficiencies of the cell types that should be present in the sample?
Well,
different data sets just don't even have the right, like some of the same cells?
Yeah, well, yeah. So one of the problems is that many of the data sets are whole cell, and so they don't include mature adipocytes. The second problem is of the data sets that are, I guess, quote unquote, complete and do include mature adipocytes, they're from different anatomical locations completely in the body. And so that's something that we've been talking about a lot, is how do we combine them, and if so, how do we combine those?
And so probably the annotations, the metadata that goes with each data set is not regularized enough for people to understand what are the limitations and about each data set.
I wouldn't necessarily say that. It's less that we don't know that. We do know what cell types are there. The problem is, and obviously for published data sets that don't have all of the cell types, we can work with that. We can, some of them have adipocytes, some of them don't. That's okay.
But maybe one of the places to look for inspiration there is actually the combined organ atlases. For example, the combined immune atlases, where clearly cells come from the liver, the kidney, where have you, and then you have a cluster that are macrophages, but maybe within that cluster you have subclustering that goes with the tissue. And as Bruce said, provided that all the metadata is coded in, it should in principle be fine to combine things, I would think, but others may have other comments.
I think at this stage, being able to gather any data that you have, even if the sampling is not optimal, the modalities are not optimal, it's okay. It allows you to then figure out the gaps. And you may have a lot of gaps, and it's okay.
So Jessica Langer, I'm from the Helmsley Charitable Trust, and we are funding essentially a Crohn's disease atlas within a larger gut cell atlas community. And one of the things we're out after for visiting here is actually to talk to other gut people. I don't know if there are any in this room. But even within our smaller Helmsley-led consortium, we're having a lot of problems with data sharing. And it's not even funding, because we have funding specifically for people to wrangle MTAs between different institutions, and it's still not getting done. So it's challenging just from a...
What is it then?
Yeah.
It's honestly, it's a COVID staffing problem, right? You can't get the right, administrative people in the universities to answer emails, because they're just not there. They're understaffed, they're overwhelmed. It's a real challenge.
Yeah.
And the other challenge that we're having that goes back to Alex's point is, and probably to Bruce's as well, it sounds scientifically like scrubbing, identifiable, information out of sequencing would be wonderful. But just, you don't have the people making those decisions as to whether or not it's acceptable are lawyers, right? And they don't understand the biology. So they see, you're going to have a DNA sequence, absolutely verboten, you're not allowed to do it. So there's very little nuance, and it's not even country dependent, it's institution dependent.
That's not true.
I'm just going to go over here.
The key point in my view is that the moment it is anonymized data, not just de-identified data, but anonymized data. You're outside any of these legal frameworks, so you don't even have to talk to the lawyers anymore, because you just anonymize, and that is the same kind of data that you would use for teaching for your students in an exercise that you would give them. And you never talk to your lawyers if you put like a exercise for students on your website with anonymized data. So this is the key point. Technical solutions that make it provably anonymized, and then you take it from there without ever going via the legal channels. Yes.
Yeah.
If you're doing a disease atlas, you can't lose your metadata.
You will use the fine-grained metadata. You lose the date of birth. If you need the date of birth, then you're screwed. But if you're fine with a 5-year age range, you're in the game.
Yeah.
They're very nice.
Yeah, Pastor.
For example, our bus system has done very structured approaches to what extent you have to broaden the granularity of your different data elements. So this is, and all of us as a large US federal entity, can be quoted. If some US lawyers give you a hard time, they can talk to Francis Collins and friends.
Yeah. So it's interesting to me. I'm going to put my head of an immune coordinator for a second, not a moderator. It's interesting to hear what the challenge you're encountering right now, because I'm at a stage where I'm even, I have the challenge of even just gathering data set. We've circulated surveys. There's the HCA General survey, the Immune Bionetwork survey, and I'm like, well, do I really have only one spleen data set? That's impossible. Do I really have only one thymus? We have a lot of PBMCs, but where are the other organs? So it's at a point where, Kerstin mentioned community engagement. It's not that people don't care. We definitely have people on the calls, but it's been really hard for us to even track data sets to a point where we're not staffed to do this. It's me after hours doing lit review to try to find data sets, which is not a scalable solution. So I was very curious with my other hat to ask how did others do that? Are you all, it looks like the lung atlas really came together really nicely, which is nice. I think immune is challenging because it's across all organ systems, so maybe that's our challenge. But I'm hearing that perhaps it's an immune specific. Actually, adipose tissue is having similar challenge.
I think also really the pandemic did not do us any favors there at all, because I really think when it comes back to, bringing the community together, person-to-person meeting, getting to talk to the people, saying, just reassuring each other that, look, you've given me data and I'm going to use it in a responsible way is really important, because every lab has postdocs that need to get publications out of that data for their career, and so there's those constraints as well. But I think we've talked for quite a while now about actually getting the data sets.
Oh, yeah. Well,
we'll
thoughts you want to add, and then I think we actually covered a lot of the second slides already.
Okay, right, fine.
Yeah.
So, hi, Clarice from King's College London, Skin Cell Atlas. One of the questions I wanted to ask, or the point I wanted to raise about collecting data, other data set, adding to your data. So we kind of bias on the data we are looking for, because we're taking data that are publicly available, adding to our data to make it stronger, but we choose the one we can take, and also we cannot take everything, because first we cannot find everything, secondly it's computationally challenging to have everything in your computer when you're doing the analysis. So one of the thing we are doing in my lab, and I wanted kind of the opinion of this room, is downsampling. What do you think about that? Because I think it's the better way to combine. So if you are taking lots of single-cell RNA data set and you want to integrate that with Spatial, it requires so much computational power that I personally don't sample all my single-cell for that. But yeah, okay, Bruce has an opinion about it, and Maria also.
Multi-set on the rare cells. You don't want to lose. You just don't want to lose any of those important rare cells. You really need as many of those as possible. So downsampling the really abundant cells that are all the same, no problem with that. But the ones that you just don't want to throw out any babies with the bathwater.
So I think we should hand over to Malte, because he's actually got experience with this, I think.
Yeah, so there are different opinions on downsampling, also in the field. I know there's like geographic sketching, which is kind of used also by the Satija lab, which is, I think, developed by the Berger lab, which they're using quite successfully. I am also a little bit skeptical in terms of rare cells, because you don't know what you're missing. Essentially, you don't know what cells you're removing to do this. We can scale data integration algorithms. Now, we recently done 8 million cells. That's enough for most cases, except for PBMC, maybe. So I guess Chloe will feel unfortunate that immune cells are a little bit more difficult because there's so many of them. But I think specifically for cases like skin, we should be able to manage that using particular algorithms that scale better. And in the future, when we map data sets on top, we don't need to retrain with all the other 8 million cells again, and so it's much, much, much faster. So then the question is, do we have this core or not?
I think the other point, in a way, that comes out of your comment, Malte, is that as an atlas is built, it's really useful to build a close collaboration with a really computational group. So somebody who really has the power to, the expertise to really have efficient algorithms, so that some of those challenges can just be overcome by good software and choices of tools.
Can I maybe add a point also to what, just to kind of reiterate what Christoph was saying before about the anonymization? There was a lot of discussion there. When you build an integrated atlas, you are essentially integrating count data on highly variable genes using some level of cell type information, if you want to supervise that. And everything else is how you use the atlas. So if I want to do SNP calling, I want to understand specific long-known coding RNAs, it's likely they will not be in my highly variable genes. And so they will not need them to build that embedding. So what you can do is you can share anonymized versions of data, as Christoph suggested, build an atlas, send it back out to the individual data contributors, where you can start calling these things and addressing more detailed biological questions to then, fine-tune your raw data processing pipelines and do this iteratively. Yeah. But I think for the initial building, and that's the kind of the crucial part, you need to seed it somehow, sharing anonymized data is probably okay.
And actually, maybe if I take the chairman's prerogative here and just to make another comment as well. We've talked a lot about how to gather the data and bring it together to build that atlas. And I think I'd really like to also throw that back to the funders. I see Jonah is back in the room. And of course, CZI has funded a lot of the networks. But the person who takes it upon him or herself to pull all that data together really is putting in a lot of time, a lot of effort, and they need funding support to do that. Time doesn't come for free. So if there are any ways of supporting these kind of activities, then that's really fantastic. And I think one of the calls that Jonah mentioned earlier is just for this kind of thing. Did I understand that correctly?
I just, while you walk with the mic, Martin, who was supposed to co-chair the session, is online. Martin, we hope you feel good. And so he wrote, I feel with you, Chloe. In the end, funding from CZI, C-Network, and the EU, the Discovery Air, allowed me to make time, as in 2 days per week for the past 2 years, just to coordinate this. So you're very lucky, Martin. And so he said collecting data for the Lung Atlas was very hands-on. Sasha, Lisa, and Malte and myself actively approached everyone that we knew had data sets, which is, I guess, also what we're doing in the Immune Bio Network. Thank you, Martin.
Great. Yes, and Martin, I think, Chloe, you just said you're lucky, Martin, but actually I think the community is lucky to have Martin and Sasha to put the time in.
Yeah, so maybe just to quickly touch, so to answer your question, Kerstin, yes, right? So the Data Insights Call, RFA, I think is one nice opportunity to begin to support collection data sets. The other thing that I would point to is, the support for doing this work in terms of, within Cell by Gene, within the DCP, right? There are resources there where you don't have to take on either the aggregation or curation, either of the raw data sets in the case of the DCP, or the processed matrices to kind of fuel some of these integration efforts. So I think there's like a wealth of possibilities there. And also I think some of the points that Malte and others have made are wonderful about kind of that iterative process to generate these reference data sets that might be different than the use of them. And maybe not to conflate those two and kind of like add to that burden or the difficulty, but to kind of like lean into some of those resources to make it easier and then also like really focus on that kind of first attempt.
Yeah, perhaps just to add to your point, Jonah, I think like we've heard from the DCP, we heard from Cell by Gene today, which was great. I guess having a bit more clarity of how we can better leverage this infrastructure, like, you know, are there data wranglers, for example, that could help some of us in gathering the data and doing, you know, lit review to figure out what has been published, as opposed to us individually trying to do this. This is not super scalable, so finding a way to kind of synergize a bit better would be really great.
Sorry, I'm Gabby Jordanova, and I represent the HCA DCP ingest service. So basically, at the moment, yes, you can definitely use our data curation teams. We're constantly on the lookout, and we think that we have this like screening of all data sets that come out. So we have different methods of, you know, well, whatever goes out on Geo, whatever goes out on, you know, people talking about Twitter, but there needs to be a closer connection, which I'm trying to establish also with, you know, even in the face of just a closer connection with the bio-network, so that we kind of know what are the data sets that we should curate, what are the data sets that we should gather this rich metadata for. But we're definitely doing this at the moment, and as Jonah said, we are doing the raw data and metadata collection. We try for that one. We have so many columns, so many features for metadata that we collect. We are also trying now to establish the curation for the flow which goes into cell by gene as well. So this is for the count matrices as well. But yeah, just I guess like closer communication, just to understand which are the data sets for us to kind of go and curate.
And am I right in thinking that you'd really like to contact people? You'd like people to contact you quite a lot prior to publication, so that you have a period of time where you can sort the metadata, and so that to throw that back to the community, you know, contact the DCP or any of the platforms really early, and they can get all your data and still keep it under wraps until you're happy to make it freely available.
And if I may ask a question back, could DCP reach out to all the different bio networks and tell us what you currently have for each of our tissue? That would be so helpful.
Definitely. I have a list of, you know, people from the different networks now to kind of get in touch with. But yeah. And we also have quite a neat flow, actually, of HCA publications. For example, when people want and write to the HCA committee, et cetera, for an HCA publication, they usually come to us, and we know to work with those contributors and curate their data.
That's great. I moved the slides along, but you know, we didn't want to stop the conversation. I skipped the previous slide because I felt we touched a bit on it, and I want to make sure we get to the data gap and data sampling. But Kerstin mentioned that it's, you know, community effort. We kind of need to bridge between groups, those that are developing new computational frameworks, those that are thinking more about the biology. So one of my questions was, you know, how are you thinking about coordinating with different computational groups? Some of you probably have computational expertise within your bio network. And so I was kind of, any thoughts on this? How are you currently coordinating? I think the lung, it came pretty naturally together, right? How does the gut do it? I'm sorry, I'm putting you on the spot, Jessica. Oh, I put you.
So we actually have sort of a support grant out to one particular group that's sort of handling constructing essentially a portal, which will allow our different researchers to deposit their data there and start with the, you know, basic computational questions there. But yeah, pretty much all of our groups have a computational expert embedded within them, and we do try very much to get those computer people talking to each other.
Yeah.
Hi, I'm Walid Khalid, so I'm coordinating the breast bio-network with Nick Navin and Kai Kissenbrook. And I think it's really interesting to hear the challenges that everyone is highlighting, because it's exactly the same that we're facing, is that whenever we meet up, we always say, well, we need someone literally dedicated to just put everything together. So I think we've come to the conclusion is that we're going to need to apply for some sort of funding, and it's great to hear about this initiative, to just literally just coordinate all the computational needs and make it in a user-friendly format. Yeah, so we're not doing it effectively at the moment, let's put it that way. So it needs a dedicated person in our groups.
That's helpful to hear. Yeah, I think it's a problem for many bio networks. And
Yeah.
And this afternoon, it is this afternoon, different bionetworks have to summarize where they stand, and there's going to be a representative from the computational working groups. They want to hear feedback, so it's really the time to tell them what you need, what are the gaps, so that they can try to, distribute resource and time to support everybody.
I would also be really interested, like we heard this morning about some really interesting tools like the cell annotation platform. I mean, how many people in the room have actually been there and tried to use it?
Well, it was only launched 24 hours ago, so I'm pretty sure.
Okay.
Pretty sure nobody went
sorry,
on
I hadn't
it.
quite realized that was that fresh off the press.
Yeah, it was.
But in that case, maybe we should just make a plug for it and say, all look at it and see whether you can find ways of actually using that to bring your community together.
That brings us to this slide. I know for the immune cells is a huge challenge because, immunologists love their labels, and so harmonizing cell label across tissue compartment is going to be an interesting challenge. So for us, such solution is quite helpful. Did any other bionetwork spend any time? I guess Lung did, but about cell type annotation, harmonization.
This is Bruce Serna. I hate to bring up this just annoying issue, but we don't. Gene markers for cells is not a solved problem. And so what people put into their maps that are, here's 1, 2, 3 genes that are representative of this cell type, they're really, well, you know what, not necessarily in all data sets are those going to be very high performing markers. And then so the best performing markers are, are gene lists that aren't necessarily clean in and of themselves, but as a group ensemble, then they work pretty well. But trying to get down to, well, what are the biologically informed markers that everybody can agree on are functional in the context of one cell type versus another, that's a hard problem. And so scaling that to the level of resolution of the cells that we're seeing right now and sometimes not seeing is, it's an important task. And
I completely agree. It is really difficult, but actually I think it's also something that's going to get easier with time, because as you've got more data with different variants in there, in a way the biological variation will hopefully become more apparent. Not
100,000% agree, but the point is we're still reinventing the wheel hours a day, every day, across many groups. And by not having a more rigorous approach to gene lists, marker lists, and annotations to go with them, and then, heads up, and if this marker's out and not very high quality, then, let's have it in front of us to share and keep, upping the ante of what's a better set of genes.
So Amanda Oliver again from Wellcome Sanger Institute. I have 2 points. The first one is just kind of more general part of the challenge, or one of the challenges of annotating cells and coming up with consensus is differing opinions. And so, I guess this is just a call for everyone to not be kind of egotistical about it, like I want to name it this way because I think, just kind of, we need to come to a consensus because it's useful for all of us. For example, for the lung, there has been, dedicated meetings. There was one in San Diego in April. And so I think this is also useful and needed, provided that it remains productive and people really come together. I think this is useful to, people agree, okay, this is the same cell type, what are we going to call it? And like a name sounds trivial, but at the end of the day, if we're all referring to cell types the same way, then it makes, the insights a lot more meaningful because it can be transferred across different papers and findings. The second thing that I wanted to mention as well is the challenge of, this is partly like annotation challenge and integration challenge, but with, for example, immune cells, sometimes you lose the granularity when you integrate because of these batch effects, which, Malta said there are benefits to having the batch effects, there are also negatives of these batch effects. And in keeping with that, I think it's important to look across networks. For example, for the lung, we have immune cells, so we should also be harmonizing cell type annotations with cross-tissue immune atlases, for example.
Yeah, I think.
Yeah, Amanda, they're very good points there. And just to add, even with, for example, again, in the lung cell atlas, we had the sort of more European-led lung cell atlas and then a hub map. And of course, there were times when very similar cells were called different things. And I think at this stage, it's not even necessary to actually agree on a name, but just put somewhere in a public space saying these two are equivalent. And actually, the coordinators of the hub map in San Diego did a really good job of that, of just saying, let's not go to the really difficult ones, but for the easy ones, let's just agree that, yes, these are the same. And I think that's already been really good for the community.
Yeah. And there's obviously the ontology working group meeting in parallel, which they are all about that. And there is, in other ontology fields, a very structured way to build consensus and to get community input, which I think we should and probably will, based on the group's output, leverage here. I think one important piece for cell annotation also is, as you already mentioned, there will be elements, vasculature, immune cell populations who are shared between organs. And obviously, we have discussed at several of our preceding meetings in Human Cell Atlas that having these cross-cutting working groups in place is important. And I think I really would encourage us to hear from these cross-cutting groups, particularly the immune group and the vasculature group, to give feedback to the specific organ networks so that we can see if specific endothelial cell phenotypes, specific immune cell phenotypes are characterized. In the kidney, we know that they change their phenotype from circulating monocytes to tissue-resident macrophages and then urinary discovered macrophages. Some of these insights might be helpful for other organ systems as well.
Regarding the cross-cutting effort, so I can at least address for the immune cells, again, switching hats from moderator to immune coordinator. So we're defining different tiers for version 1.0 for assembling our atlas. The first one is focused on immune tissue, but the second was actually going to entail coordinating with the different bionetwork to extract any immune cells that have been gathered so far and try to work with every bionetworks to harmonize labels. And I'm going to use that time to make a bit of an announcement. The Immune Bionetwork is trying to organize a cell annotation jamboree because we realize that we all annotate perhaps very differently. And by annotation here, we're not talking about slapping a cell label on a cluster. We're talking about if we give everyone 3 million T cells, how many subsets do people discover? If we have exactly the same T cells that have been integrated exactly the same way and we give everyone the same data object. And so if you're interested in joining, we're going to be launching this Google questionnaire for those that want to sign up for it. And this week, the first jamboree is going to be PBMCs, because if we cannot even agree on PBMCs annotation, there's a bigger problem out there. And so we felt like this would be like a good place to start. And this is building from an integration workshop that CZI launched recently. So it's COVID-19 and normal PBMCs. And so it's going to be where you can join anywhere you are in the world, because it's going to be virtual with very defined tasks. And then we'll kind of try to regroup at the end to compare our results. So just like a shout to try to tackle this challenge. And
I would love to understand there, so specifically for the example of the, I think, is it kidney? How you use ontologies to seed consensus. Because, so for example, for the lung, it was not always easy to start with a prior ontology to then build off of that, but instead we found it quite useful to just kind of be able to get a bunch of people's opinions and then see whether those opinions we can put in a structure and then later worry about adding that to an ontology. How do you seed the consensus with something that has prior structure that maybe not everyone agrees on?
I think ontologies have a mechanism to build consensus. That's what I implied. So you post, this is a structure, this group has who met on a specific theme with a limited community, this is our proposed structure. And then the OBO community has a formal process where that proposed structure is posted for larger community input. What you want to avoid, that people meet at Vienna, have a good few classes of Zweigel together and come up with a final solution for the world, and then push that forward. and are becoming exclusive. And I think that is a real concern, and Human Cell has been very good in being that open community and leveraging some of these tools to that extent. Yeah.
Perfect. Yeah, that's exactly what I was looking for.
I just would have a practical question to that. So we also have gathered a community, like for different structures of the skin. And when you have small groups who ask to give you feedback, how they actually would call things and according to their expertise, how do you practically share? Do you make a little tool for them where they actually can query for genes beforehand, not to just send them the Excel files that they have to scroll through and search gene by gene for subclusters?
I think it's a tool we heard earlier this morning, I think is specifically developed to do that, and I'm really hopeful that our network and others will be able to do that. One additional feature which each community should think carefully about, this is not all about single cell, single nuke and mica genes. They only go so far. I loved what the brain people do, that you have the functional assessment linked on a single cell level to your signatures. And in each of our domains, there are unique cellular features which should be considered to be brought in and mapped. Kidney, obviously, its specific transport capabilities, which we know after a couple of 100 years of renal physiology where they live. And integrating these type of physiological characteristics to the cell population stratification is important and really is a unique feature. Each organ system should creatively think, and quite often you have to mobilize expertise from the 60s, 70s, and 80s to really bring that knowledge back to our now cell and molecular-based redefinition of cell population.
Do we have comments virtually? No?
Yes, so we had a question regarding different ethnicities, in particular Africans, but maybe we can consider that a little later, or shall we have that discussion now?
Sure. I mean, I'm trying to track time, so we have about 20 minutes, and so I'm going to highlight some of the key other points that were open for discussion so that people can jump around.
Yeah.
But
so
diversity questions about sampling.
Yeah.
So I'll quickly summarize the other thoughts so that then you can jump to any topics. We wanted to know whether you saw any opportunities where we could, better intersect. So, we talked about shared immune cells, harmonization, shared cell types across tissue to harmonized. But if there's, any needs in terms of sharing more the SOPs, computational frameworks, experiential framework, if there's any tools that you feel is really missing that could really help you build version 1.0, that's screaming at you, aside from support, which is obviously very key. I want to hear about it. That's the missing tool part. And then somebody asked earlier about data gap. So, how are you assessing your data gap, your sampling strategy, and so forth to kind of help build version 2.0. And I'd love to hear about what are your metrics of success for version 1.0? How are you assessing that, your version 1.0 is actually going to be successful or useful or any metrics? So to summarize, any missing tools, technologies, method, SOPs you think we could share? I have one on top of my head. A lot of us have to figure out how to move to spatial. And yes, of course, we need to optimize our own specific tissue handling, but there might be broader tools that could be shared more broadly so we don't reinvent the wheel. So opportunities, missing technologies, data gap, how are you assessing data gap, and metrics of success?
Yeah. So actually, because we want to include the online audience, and there has been this question about other ethnicity which fits into the data gap, maybe I can just take a survey of whether there are any groups here who are actually looking at different ethnicities and what efforts are being made.
I think in kidney we obviously have the advantage that we can access live tissue donors for our tissue, and certainly in our NIDDK-funded Kidney Precision Medicine project, our recruitment is specifically targeted to reflect the ethnic diversity, at least of North America, and we are on target for that as of now.
Yeah. Any comments here for ethnicity at all? Marco?
Hello, Marko Nikolic from UCL. So yeah, we've got a CZI-funded grant looking at taking upper airway samples from various ethnicities, and we're basically taking advantage of the fact that we're in London, multicultural London, and specifically targeting ethnicities from which we haven't collected yet.
So perhaps that's a call also for every bio network for a specific organ system to coordinate with the CZIC network that's supporting ancestry-based effort. So for example, for the immune bio network, we're trying to coordinate with those that are developing immune-related data sets so that we can start integrating and harmonizing.
Just one final comment. Obviously, capturing ethnicities in Europe or North America is not reflecting the diversity of our planet appropriately. And really kudos to CCI to reach out and have touch points established in South America and Sub-Saharan Africa. And what would be helpful is, as tools are developing that tissue can be shipped and harvested and potentially analyzed, linking out in the different organ systems to some of these sites who tissue procurement can take place could actually add critical diversity, which we are otherwise missing.
Just to add to that, I think with those efforts, it'd be really good also to transfer the expertise to actually analyze samples at the site of collection rather than...
And CCI and Human Cell is closely linked with the H3 Africa effort, who has done a really good job in allowing Sub-Saharan African investigators to drive these efforts. And please continue to reach out. And if some technical expertise is needed, how to transfer tissue effectively around, I'm happy to help, as we have aspects, infrastructure in place there for quite a while.
Thank you. I'm curious to hear how others are evaluating data gap. Like who has... Like I speak for the Immune Cell Atlas. We have a lot of RNA and CiteSeq, a bit of a repertoire. Don't really have multimodal, except from 1 or 2 data sets. We have almost no spatial omics. In terms of sampling strategy, I can speak for the Immune Bio Network. It's pretty limited in some cases. Aside from the HubMap data set location that have not always been recorded, it was mostly like a biopsy. So there's a lot of gaps in terms of capturing the full spectrum of immune cells. But I'm curious about other bionetworks. How are you thinking about your data gap? How are you tracking it? How are you thinking about figuring the gap moving forward for version 2.0? Any thoughts about this? I mean, I realize that we may not be funded for this, but the first step is to figure out the data gap so that we can get organized and prioritize what are the data modalities we need to prioritize to improve our next version.
So I guess I can speak on that also from the perspective of the gut, which I work on, as well as lung and mucosal immunity. For example, there's also gaps for large organ systems like the GI tract. There's gaps in certain locations that are undersampled. For example, for the gut, there's not a lot of healthy stomach data out there. And so these are also considerations that we need to make and become obvious when you start to integrate and collate the data.
So here, you're not even talking about sampling within a certain tissue. You're saying there's a whole tissue that's missing. Yeah, that's a big gap. Yeah.
Maybe I can say from the perspective of the lung atlas. So we're actually using the version 1.0 of the Human Lung Cell Atlas to identify where we need more data. Yeah. And so this is specifically, I think, something that was brought up on Twitter quite poignantly by Lyra Proctor, that it's mostly adult white males. And so one of the aspects there that we've been trying to reach out to different groups who might have more diverse data sets, but the additional complexity is that when we have ethnicity, how do we encode it? So typically that's been self-reported, and the encoding there is very different across different groups. And something we're trying to do there together with Joseph Powell, who's also involved in the SEED network, is to build ancestry calling pipelines from single cell data, and we don't know how well that works yet. Hopefully well enough that we can do this, kind of roll this out across the network again, and then assess exactly where we are in terms of representation of diverse ethnicities.
Yep. And so how are you thinking then about number of donors? Because you can also use a data-driven approach to try to figure this out, right? You can try to, I mean, at least I guess for us, for PBMCs, there's so much of it that we can downsample and ask, how many donors across different diverse background did we need it to identify all existing cell subsets? But we can do this because we have so much data, which is not the case for most other tissue. So how are you thinking about this?
Wait.
If you get results from that study, that you share that with the other organ systems where clearly sampling is a lot more challenging, so that, ceilings are established, at which stage can and cannot be claimed.
Yeah, we'll definitely share. This is one of the tasks for the Jamboree. We're trying to use a data-driven approach in terms of, we have the Asian network is contributing unpublished data, as well as some others in Asia Pacific. Same thing for gender, right? Like some folks have suggested for immune cells that you need more female than male, and we'll try to use a more data-driven approach to ascertain this. And so, yeah, of course, we'll share this.
I'm actually going to go to the online first so that we can...
Thanks. So the original question from about genetic diversity came from Mandeep Kaur, who's just replied and said, please can the contact details of anyone coordinating tissue sharing from Africa please be shared, please?
So, contact for tissue
details. Contact details of anyone coordinating tissue sharing from Africa please be shared.
There's a comment here.
Feel free to email Jonah.
So just a quick follow-up point on that. I know the equity working group, and their contact is on the Human Cell Atlas website, is doing a lot of work to coordinate both, of course, things like enabling PIs in the global south to perform research, but also develop kind of metrics to represent things like, or at least there's been discussion of it, I don't know if it's moved forward, to represent like ancestral diversity in the sort of Cell Atlas effort. So I wonder if some of these questions might be good to field out to the equity working group, and also if there might be room to sort of, for PIs who are addressing these questions to cross-pollinate, just discussions with that working group. They're doing a session, I think, on Thursday, but I realize it's outside of the main meeting, so I don't know if everyone will be able to attend, but I know they're doing something Thursday.
Great, thank you. Yeah, Malte.
And then, okay, go ahead, and then we'll go to you.
The question you asked before, Chloe, was about basically when are we done, right?
When we're done, and also I guess that's the
10,000.
But let's say version 1 versus version 2. I was going to ask you about also modalities. How do you figure out modalities?
So maybe first, this is a really difficult question. Like, how much do we need to identify that we don't have any more gaps in data? Right? It's essentially how are we done. And there's multiple different levels which you can answer this question on. I think in terms of the quantitative ways, also from what you're getting out of the current atlases. If we're talking about identification of cell types or particular states, it gets a little bit easier. What do we have now? What's the proportion of the whole cells? And then kind of seeing, would we capture that with a certain number of individuals or of samples? The more difficult question is, what do we do with an atlas? Like, if we build models on top of an atlas, let's say to model patient variation, we are nowhere close to done, because 100 patients or 100 individuals is not enough. There's always going to be more more things you can get out with more patient heterogeneity, and I think that's kind of an open-ended thing. I think we're quite far away from having an answer there.
We've got someone over here.
Hi, Giovanna Prout. I'm from Industry, Deep Cell. I'm curious to this group, there's already so much of a data gap in the current modalities known, but what is the appetite for new modalities, more unfamiliar data types, let's say quantitative morphology?
Any thoughts about other modalities?
I mean, if I can maybe just comment. So we're certainly very interested in going towards quantitative morphology, and I know CZI is as well, because they're, for example, funding sort of hierarchical computer tomography projects that look at very hoping to go for single cell morphology. So I think the appetite is huge, but the technology is maybe not quite out there to reach a wide audience, and it's really still at the stage of doing small pilot studies to see how much resolution you can get, and then also whether you can use those same tissues and also get single cell expression data for the same samples so that you can then start to think about combining the two.
So we actually had a chance to do a little bit on COVID, a look at just African Americans and Hispanics were at elevated risk of acute phase severity. And so we tried to correlate that with ACE2 expression in different tissues. So in trying to address that kind of a problem, and so we used GTEx data and then the available lung data too, and as Malti said, it was very underrepresented for African American. GTEx is better for that, but on the other hand, at that stage it was all bulk sequence, and then you deconvolute and all that stuff. What's useful? What do we need to really have enough of a reference that you can do queries like that to see, is there a tendency to have really altered proportions of these different cell types is the key question. Is there elevated ACE2 expression in them? No, but in submucosal glands, there's something very interesting, different in terms of the proportions of those anatomical substructures, and those drastically change the physiology of the system. So that's the kind of level of knowledge that we need to be able to have at least enough power into different populations so that we can see, is there a correlation to differential disease risks, and then work on it.
In terms of integrating like organ morphology, obviously a lot of work has now been done, population scale MRI studies. Even a subset of the UK Biobank has been studied with kind of body imaging efforts. There have been extensive data sets, even across diverse populations of brain imaging, of heart imaging, driven by kind of the field of epidemiology, also studying human diversity in these organs and doing GWAS for organ morphology. Obviously, what the weakness of these studies is that the molecular dimension is missing. It's basically the long association from a SNP to kind of a structure in the brain, which loses a lot of the biology in between. I guess what is the strength of the human cell atlas, that it is really the strong focus on the molecular basis of cells. And where we can do that, have kind of functional imaging data, but with a molecular basis, that's powerful. If we now think we can kind of reinvent kind of 3D imaging by giving up the molecular basis, there's so many other area of fields that have done this with perfection for the last 20, 30 years that we should not look too naive in this regard. But often these communities that have been doing this for the longest time, they have either have not having any molecular information, or they had traces that give you 1 or 2 targets, which is powerful. But it doesn't give you kind of the depth of the picture that the Human Cell Atlas type methodology can give you.
I guess you're getting towards that common coordinating framework indirectly, which was one of my other questions, because I'm curious how people are organizing around this. Some organ systems have more structure, some others don't.
We have to wrap up.
No, we have 20 minutes.
Oh, okay.
And then Jessica.
I think one advantage is if you have biopsy access to tissue, then you obviously can couple non-invasive imaging procedures with tissue sample at the same time, and you then can align the digital pathology or anatomy with the imaging. In the kidney field, we are doing this across reasonably sized cohorts, now both in the US and in Europe, where biopsies are obtained, usually not from healthy individuals, but from deceased individuals. But nevertheless, some of the interdependencies, oxygen tensions, different regions of the kidney and how they correspond to structural features, renal plasma flows, and the associated molecular signatures are data sets which are emerging right now. So these are elements where this is possible. NIDDK is hosting NIH a workshop next month to actually discuss how to ethically work with volunteers to obtain tissue via biopsy procedures and what orthogonal data one potentially could capture. In a pilot project, this is happening indeed already, even in the kidney field. So there are opportunities coming along where we will have very small, hopefully nested cohorts where the multi-model data point comes then in play, that even if you have 10, 15 individuals where you can do the crosswalk, that might allow you then to disambiguate some cell population or to link specific structural features to specific molecular events where you then can project, for example, in the UK Biobank fMRI data.
Jessica.
I had 2 comments. First of all, one about where the data gaps are, and I think that goes to biopsies, because you biopsy a tissue, you think you've covered it, but you've really only sampled the epithelium, especially in the gut. They just do the epithelium. You're missing the full thickness, which is a tremendous amount of information.
Yeah.
So that's one comment. And then for the common coordinate framework, yeah, that's something we are in fact working on quite extensively in the gut. The problem is that the gut is very, very large. And it's highly variable from person to person. So the best we can do is to, find landmarks along the way and say, how many centimeters are you from this landmark? We really don't have a grasp on how much, for example, the ascending colon changes from the bottom to the top. Like, we don't know if sampling from, the top of that area and the bottom of that area are going to be equivalent or not. These are part of the data gaps that we have.
Yeah.
Currently,
and
especially in very large organs.
And I think, the common coordinate framework that you brought up is a really important thing for all the different atlases. And I think we're all still struggling with that, to be fair. In the lung, we are relatively fortunate because you can, of course, count the bronchial branching. But of course, also, often when you sample, you don't know where you're sampling. And so even though that should be possible in theory, it's much harder in practice. Might also be worth pointing out that there's another session, I think led by Katie Berner, on the Common Coordinate Framework, and they're putting together these ASCT tables. So people who are interested in that should go and look that up.
I think that'd be great, because I have to admit that for the immune system, it's not really clear to me how we establish this, given that lymph nodes will change across individuals, across location within an individual.
But then there are organs where there's clearly already established immune zonation, like across the kidney. So
that's correct.
As the different organs begin to grapple with their own CCFs, then I think you'll be able to.
We have our own immune cell location within the common coordinate framework that we're working on in gut, for example.
So perhaps that's the solution for the immune bio-network, working with every tissue, though we have to figure out our own for spleen and thymus and so forth. We have about 10 minutes. I'd love to hear about either other, I'm surprised I was waiting to hear about other modalities. I'm wondering how people are thinking about like combined measurement. Is it even worth the cost? Because there's just the reality of funding and, co-measurement of RNA and ATAC is twice the cost. So for CiteSeq, you add a lot actually on sequencing costs. I was curious to hear how people were thinking about modality integration and how this would improve their annotation and how you're even thinking about incorporating spatial transcriptomics, because I think across most bionetwork, a lot of us don't have a lot of spatial transcriptomics for this first version. So do any of you have conversation about this? Or just very focused on the RNA space?
Hi, so Clarice, King's College London, Skin Cell Atlas. So we've been working with spatial transcriptomics recently with 10x Visium and in situ sequencing. And it was really informative to see the RNA level in the section. But I think so far what is the most informative for us was to integrate it with the single-cell RNA-seq, because you can really project your cluster and that give you more information about the annotation, actually, because you know well your structure of the tissue, so you kind of understand better. But something I kind of wanted to do and I'm a bit struggling with is to integrate everything together. So single-cell with 10x visium works to a certain extent because we don't have single-cell resolution, so you kind of see the spots, but this is kind of give you nice co-localization where some cluster are close to each other in a tissue. This is working really nicely with in situ sequencing in the skin with our single-cell so far, but putting the non-cellular resolution spatial, so 10x visium, with the in situ, which will be super complementary, because in a hand you have the whole genome, in another hand you have your selected set of genes, so 200 in our case. It's really challenging. So I don't know if some of you have some thoughts about that.
Tom from Sydney. I guess for the immune bio network, so much of modern immunology is run on sort of, FACS or CyTOF or spectral cytometry now, where having site-seq as a part of the picture is so important. Because I guess not every experiment in immunology will be single cell, which has this kind of greater feature range, but having something that you can kind of use to translate between them. And we do this quite a bit now, because you can sort of ignore the RNA for a bit and lean on sort of a couple of hundred site-seq antibodies, which then give you some new insight into how to tackle a complex disease situation. I think that's very important, because that helps them with scale, because if we can run, hundreds of millions of cells in FACS or spectral fairly cheaply, which then means you can have greater patient cohorts and things. So I think that for the immune network is maybe a bit different to other networks, because we're so dependent on proteins for most of our work. Yeah, very, very important in that context, I think.
Maybe I can also comment on the spatial transcriptomics for the lung, which we have done quite a bit in our lab. And actually, it is very, very powerful data. It obviously really helps to have the really well annotated version 1 of the atlas that you map into that data. But you can really use it very powerfully to validate cell types you've annotated. So if there may be 2 fibroblast types that look quite similar on the UMAP, so if you then have the spatial data to see that they're actually mapped to quite different places, that's really good data to say that, yes, these really are different cell types. And for that, you don't actually need single cell resolution. Your 50 micron spots are usually quite sufficient for that. And we also see, new structures. So we've described this immune niche for IgA plasma cells at the submucosal gland. And then having, just that visual impactfulness of seeing a plasma cell completely co-localizing with a submucosal gland cell, I think is really powerful.
We'll go at the back and then come at the very back. Yeah, go ahead.
I just wanted to add something on that. That's true that the 50 micron resolution is really powerful, but I think it depends really how the tissue is structured, because for the skin, for example, in the epidermis, 50 micron is actually, you cannot see all the different differentiation stages, impossible. You're going to see undifferentiated and differentiated keratinocytes, and that's it. Yeah. So, but I'm pretty sure for the lung it's really good. And I just wanted to correct, it's not challenging to put in situ sequencing with single-cell RNA-seq. I was talking about the 2 spatial transcriptomic method, 10x visium and in situ sequencing.
Oh, got it. I'm going to find it.
Sorry.
No, please do correct me. We have scribes that probably got it better than me also.
Hello, I'm Maria Llamazares from the DECAIF set in Heidelberg. And although I'm a super fan of single cell, of spatial transcriptomics and bringing cells into context, I think that we also have such a depth in data from bulk data sets such as TCGA. So now we have our single cell lung tumors, but we don't understand much because we only see superficially, okay, we have all these cell types, we have all these tumor cells, but what else can we extract? So I think we really need to go back with our single cell data into TCGA or larger cohort data sets to understand a little bit better what's happening.
Yeah, that's a good point.
Yeah, but I think there was a really good example in Mars Hannover's talk yesterday where single cell data was used to deconvolute certain childhood tumors and then seeing quite specific tumor subtypes. So I think it...
And indeed, Malte, Herbert, and Fabian, they have done a super job also in fibrosis with such things.
So we have 5 minutes left. I guess, I guess I had 2 questions. I guess one is building what you just said. How are different bionetworks thinking about disease integration for version 1? I think, to put some nuance to this, the immune bionetwork goes back and forth about including disease because we think disease is needed to capture the full spectrum of immune cell state. Otherwise, you're going to be a bit limited. So curious to hear any thoughts. And then I want to see if anybody wants to volunteer metrics of success and utility. Maybe you want. No, you don't want?
Just when you put multiple COVID data sets together and you look at the different compartments, and I'm saying this about your proposed PBMC immune atlas, it's really flat. I mean, you're not seeing incredibly important things, and you really have got to take on the disease states and the other in the compartments. So
can I cite you, Bruce, next time I have like a session?
Anytime.
Can you come to our bionetwork? I couldn't agree more. I think if you really want to capture the full spectrum of cell state, you need some, especially for immune cells, you need some perturbations. I don't know if it's the case for every tissue. It's probably the case for every tissue, although it probably gets tricky for other tissue types in terms of injury and so forth.
Hi, I actually just had a question. This is Lisa McGinnis from Genentech. I was wondering what people think about normal adjacent. It kind of goes into your idea of capturing the entire spectrum, and that's both true for tissue and for immune cells. And so it's often easier, as a pathologist, we often will get normal adjacent tissue a lot, but it is different, so it's not your normal tissue at all. And so what are people's thoughts? I feel like it could be most useful for immune, but also for other tissue types.
That's a good question. So the question is related to tissue sampling, tissue adjacent to tumors.
Well, I could tell you within the context of IBD, it's been shown that if you're like adjacent to an inflamed area, you already have inflammatory changes. So we can't use that. We talk about involved versus uninvolved, but
yeah. I think as other will chime in, I'll chime in as well. I think the important thing is to have good metadata to go along, because the same argument can go from those that do tissue procurements from autopsies. Obviously, if you passed away, you're not normal and healthy, or unless it's a car accident, but even that, right, you can have all sorts of cytokines, storms, so good metadata that tracks with the tissue that was generated is kind of key. And then as we assemble, then we can probably figure out what's normal and what's not. Oh, behind you, if you don't mind passing the mic behind you. And we'll go at the back for breast.
Yeah. Lead first.
Yeah, so in our atlas, we've got some contralateral normals. So women that had breast cancer in one breast and sequenced the other, removed the other breast, which is supposedly normal, which actually doesn't look that normal, to be honest, because it's probably the inflammatory response. We also include germline mutation carriers, prophylactic mastectomies. So this is how we're approaching the disease. I think the tumors themselves, we haven't touched that.
I was going to raise the point that, there are quite unique challenges in getting healthy tissue sometimes, even, if you get healthy tissue from organ donors like we do for the lungs were rejected for transplant for some reason, so there's always limitations there. Also, there's no perfectly healthy human being. We're all a spectrum of health and disease, right? And so I think having things like adjacent normal or whatever kind of best next thing that you can get with integration with maybe more healthy and better quality organs, then we can get this spectrum from health and disease. I think it's super valuable to have, adjacent normal or non-inflamed or what have you.
And really continuing along those lines, establishing that spectrum is helpful, and even if a sample size is minimal and would not be sufficient in itself, it might help you to identify that if you take living donor protocol biopsies, they still had anesthesia and have an ischemic response. So if you get unaffected parts of tumor nephrectomies, that is even more aggravated. But you might get a few biopsies who show up normal histology and a different cause of the disease is found. Or, and this is really an active discussion, Now, are there volunteers who are donating tissue for research? And this clearly gets into an area where with the ethic boards one has to be very careful where and how to consider such a donation. And that's just one final comment. I really want the group to also think. Right now we are talking about data, derived, and all the elements. These come from human beings, some of them living, some of them deceased, but they will have first-degree relatives. And do we think about how and where we can share some of the informations and insights we generate back to our tissue donor or the next of kinds of the tissue donor? That gets also back of the earlier discussion, as we are potentially detecting, germline mutations who have an impact of the donor or the next of kind, the return of results discussions. So these are aspects which really I would recommend you consider early on and talk with the ethics group in Human Cell to make sure you are communicating. And as a network at large, having a segment of our web presence targeted towards people who donated tissue to our efforts, I think would be adequate. And
it helps with engagement, right?
And actually, that's a really good point. And actually, one of the aspects of the Discover grant was also to have a public outreach, to have patient engagement. And there were a couple of workshops that involved those groups, and it was actually really, really helpful to hear what they wanted to get out of an atlas. And I think it really helps both sides because it informs the sort of patient base about what might now be possible. They might just use it in a very different way from the way we look at it, but just see, where would those cells that are diseased normally sit? Or, just sort of things that you could possibly get from a textbook, but they find an atlas actually quite useful.
I would recommend, if you have a moment, go to the KPMP website. We have patient participant testimonials on there. And for us, it was transformative to have patients as part of our governance and steering committee. A lot of internal scientific discussions become quite, theoretical when you have people who are suffering of real diseases in front of you. So it has been a very healthy experience and really changed a lot of what we do and how we do it.
That's great thought. We'll take one last comment and then we'll wrap up.
So super short, and coming back to this normal adjacent and so on. So are we thinking in integrating also some histology images of the donors in our atlases? Because then it's also very useful to see, okay, here we have the normal cohort. Now I want to exclude, for example, because I have more experience in this or that, I want to exclude all these donors because histology is looking like that and it's telling me there is certain field cancerization. Would that be possible?
I think it will be possible. I think it's definitely in version 2.0, maybe version 3, because I think everything we've talked about today, and maybe this kind of can lead into our wrap-up here, is that obviously at this stage, I think it's really important that we actually bring together all the information that we have into a version 1.0. And we should also not be so ambitious that because it's so complex, we don't get anywhere, but just take the single cell data that we can integrate and actually put it on a portal where, people can then, where the community can go to it, and then there can be a conversation, that there can maybe some use of all those platforms we heard about this morning to improve that. And then as we're moving towards the version 2, we're thinking about the gaps and the new technologies. And I think one of those gaps is definitely good morphological data, H&E sections, that I think would also really speak to the whole clinical community, because that's, of course, what they see all the time.
So thank you all for your participation. And I realized we didn't figure out a way to gather all of those. I tried to coordinate bionetworks, but if you'd like to gather over coffee break so we can exchange tricks and ways of engaging our members, please find me. Also, nobody gave me examples of tracking for utilities or success, so if anyone wants to come in front and tell me, otherwise I'll make them up. No, I won't make them up. I'll put some of the ones that have been submitted. Thank you again.
Yes, thank you for your participation.
Thanks.